Good afternoon, everyone. I would like to thank you for joining us here today. My name is Jason Gerbino. I'm an applications chemist with Milestone. I'm also joined by Maggie Castellini, a researcher at the University of Alaska Fairbanks. Today we'll be talking about direct mercury analyzing by the DMA-80 and how that applies to the mercury analysis of fish, animal, and biological samples. So what we're going to talk about today is briefly would like to tell you a little bit about my position and what I do here at Milestone. I'll tell you a little bit about Milestone as a company itself, some of the current techniques used for mercury analysis, why analyzing for mercury is important. Then I'll tell you about direct mercury analysis and how our system works and some of the functions that make it a very good asset to bring into your lab for analyzing mercury. We'd like to end the presentation with Maggie Castellini telling us about what she does at her lab, who she works with, and the types of analysis that she does. It'll be very interesting, so I'd like to thank Maggie for joining us as well. So my name, again, is Jason Gerbino. I'm an applications chemist here with Milestone. I work in our North American headquarters located in Shelton, Connecticut. We have a full applications lab here, which is where I work. So most of my time is working with users, whether that's remotely in my lab, where I work on method development and helping optimize different methods for our user base. Our global headquarters is located in Bergamo, Italy, and we have all of our manufacturing done in Germany and Switzerland. We have over 20,000 labs using Milestone products in over 65 countries worldwide. Our North American headquarters, though established in 1988, our company was founded in 1988, so we've been around for some time now. In our North American headquarters, we have a full-service customer support department with a fully stocked inventory for all parts and consumables that any of our users would need at any time. We also have our service support department here, located here as well. And we have our service engineers located regionally so that no matter how remote our users are, we have a service engineer located fairly close to their location so they can come out on site and provide on-site service as well. So just a brief rundown of our direct mercury analyzer. So our first machine was released in 1992. Since then, we are currently on our 6th generation of analyzer. So we've really optimized the system itself, resulting in a very stable and very reliable system. We have over 2,000 units installed worldwide, and that's like the highest install base for any direct mercury analyzer. We were also fortunate enough to have been referenced in EPA Method 7473, which is the direct analysis of mercury by thermal decomposition and amalgamation. So as I'm sure everyone here that's analyzing mercury currently knows, mercury is a naturally occurring element, and by one way or another, be it natural sources or human sources, mercury can tend to get into our ecosystem and then eventually enter into our food chain. So the health risks of mercury are pretty well documented, but it's some pretty toxic stuff to come in contact with. So making the analysis and detection of mercury a worthwhile scientific goal to determine where exactly in our food chain and ecosystem mercury may be contaminating. Some of the conventional techniques for mercury analysis are generally by cold vapor atomic absorption, or ICP-OES, and more recently ICP-MS. So these two techniques, what they do have in common is they both require a sample preparation step. So this is most commonly going to be done by some sort of acid digestion, where a concentrated acid is heated with the sample on a hot plate or hot block or in a microwave. This is a very time-consuming process. It involves the use of concentrated acids, and depending on what type of lab, for academic labs, for having a sample prep step in the detection of mercury, you will have to take concern for operators coming in contact with some concentrated reagents. This can also be a quite costly prospect. Generally, depending on what your detection limits are, high purity acid is generally used in the digestion step. So that alone is pretty expensive when you look at the high purity acids, but also generates a lot of aqueous waste because you're using several volumes of concentrated acid, so that needs to be disposed of after your analysis. So this, again, takes some time. Even in a microwave, it takes about an hour, but I've seen some hot plate or hot block methods that can take 3 to 4 hours upwards to a whole day for the sample to be completely digested. Once you get to your analysis step, analyzing mercury specifically can be a little bit troublesome because mercury has a tendency to stick around inside the machine. So that can also lead to some memory effect and contamination of the lines, which creates a whole other set of problems when you look into just the upkeep on the machines for analyzing mercury itself. So where a direct mercury analysis differs is, first, there's no sample preparation in terms of a digestion. So your sample can be placed directly into the system and analyzed. So you save a lot of time on the sample preparation step. So there's going to be no acid digestion, so you're not generating a large amount of acid waste, and you're also not having to purchase a lot of those acids used in the digestion process. The system itself works on a very automated basis due in part to the software, so that allows you to load up your samples into an auto-sampler tray and hit the start button, and away you go onto your analysis. As I mentioned earlier before, we were fortunate enough to have been referenced in an EPA method. So there's an EPA method approved for the direct analysis of mercury in solids and waters, aqueous solutions. This method is EPA 7473, was introduced in early 2007, so it's the thermal decomposition and amalgamation and analysis by atomic absorption spectrophotometry. The detection range is 0 to 600 nanograms with a detection limit of 0.01 nanograms. So for the any labs that are looking to comply with any EPA methods, there is an approved method for the use of a direct mercury analyzer. So here's a schematic on exactly how our system works. Starting from right to left, we use a carrier gas. So you can see from the oxygen flow regulator, you can use oxygen or compressed air. Your samples are loaded onto the 40-position auto sampler tray, where they are carried into the decomposition furnace. In the decomposition furnace, the sample is completely combusted and volatilizing any mercury that is contained within the sample. The carrier gas then moves the mercury and all of your other sample contaminants into the catalyst. And the catalyst is the heart of the system here. There's two functions that the catalyst has, the first being to trap any non-mercury contaminants from passing into the spectrophotometer cell, but it also reduces all the mercury species to elemental mercury, Hg0. The carrier gas moves the mercury from the catalyst chamber onto a gold amalgamator cell. So the mercury is collected during the entire decomposition process, and once the decomposition process is completely finished, the software sends a signal to the amalgamator where it flash heats to over 800 C, simultaneously releasing all of the mercury from the sample as it enters into the spectrophotometer cell. In the cell itself, the mercury passes through two detection cells where we have a dedicated mercury lamp that is tuned to a specific wavelength for mercury. The height peaks are detected by the system, and a readout is sent to the terminal from the system so you can determine what level of mercury passed through the detector cell. So the samples are loaded onto the auto sampler tray. We offer two types of boats to contain your samples to load into the tray. The most common type of boat that we have is a nickel boat. So this is going to be perfect for your samples where you're analyzing any biological tissues or plant samples. So the good thing about the nickel is they have a very, very high lifetime where they can be used over 2,000 times in the system and still maintain their complete integrity. We also offer the boats in quartz, and we generally only use the quartz for acidic or aqueous solutions. We also use them for calibrating the machine as well. But for all other types of applications, solids, soils, the nickel boats are what we recommend, and they also are very inexpensive. So they can be small, so if they do need to be replaced, there's not a huge cost associated with replacing the nickel boats. So the catalyst and the amalgamator form the heart of the system, as I mentioned earlier. So currently we're on our 7th generation catalyst tube. So we've been able to develop a very stable and reliable catalyst that is capable of producing some very reproducible data. Again, just to recap, it will trap all the impurities and convert all the mercury species to elemental. It's then passed through to the amalgamator, which collects all the mercury and then releases it all simultaneously into the detector cell so that you can analyze all the mercury contained in your sample all at once. So to show the efficacy of this system, we compared 3 unknown fish tissue samples and 2 SRM samples compared to cold vapor and ICP analysis techniques. So we use DORM 2 and DORM 3 as our quality control SRM samples. So just to refresh what the accepted concentrations for DORM 2 and DORM 3 are, DORM 2 has a concentration of 4.4 plus or minus 0.8 ppm, and DORM 3 has a certified concentration of 0.355 plus or minus 0.056 ppm. So our results are very comparable to the conventional techniques for the analysis of mercury. And the last part of the system that I didn't mention in the schematics, but after the mercury is carried through the spectrophotometer cell, it is collected on an activated charcoal trap. So all of the mercury that's released from your sample and analyzed is then collected on this charcoal trap. So there's no waste generated by the system that is not collected, which means you don't have to have a dedicated hood space to vent the system into. All of your mercury is collected on this charcoal trap, and they're replaced about once a year. What this also allows you to do is be mobile with the system. So if your research involves any field work, the system can be brought out on site. And because you're not generating any mercury waste that you can't dispose of at a later date, the trap makes it very easy to bring the system with you without having to worry about contaminating your field site. We offer the DMA in 3 different configurations. What's going to be most common for your type of application, again, for biological fish and tissue samples, is the dual cell. It's a lower detection limit of 0.003 nanograms and an upper limit of 30 ppm. So it has a very dynamic range and a very common configuration that we offer. For those that sometimes occasionally do wastewaters or very, very low concentrations of mercury samples, we offer it in a tri-cell configuration, which contains the same 2 cells as the dual cell. However, we put another cell in the front end of the system to lower the detection limits to 0.001 nanograms, which puts its lower limit at 0.1 ppb. So it tags right along to the dual cell by an addition of a low limit cell. We also offer in a wide range system, which allows the analysis of very, very high concentrated samples, upwards of 700 ppm. So this is similar to the dual cell in the first low-range cell, but we add a much higher range cell onto the end of the system to be able to handle such a high concentration of mercury. So this is most commonly used in the mining industry, where they can be coming in contact with such a high concentration mercury sample. Again, the most common type of configuration for tissue and biological samples would be the dual cell configuration. The system is controlled from either color touchscreen terminal located in the picture here, or it can be completely controlled from a laptop PC. Both would run on the same software. It's fully CFR compliant and allows for multiple user access levels. It also records any logins or changes made to the system as part of that CFR compliance. It has several USB ports allowing you to connect any keyboards or mouses to make it a little more user-friendly, and also will connect to some of the more common balances to allow for your mass inputs to be done as easily as possible. One of the key features that the software has is an auto-blanking feature. So this allows a user-generated upper and lower limit for how clean the system should be before continuing an analysis. So if you're running a variety of samples, you have some higher concentration fish samples and you have some lower concentration plant matter or soils, and you can run them all in the same analysis cycle. And by defining this upper and lower cleaning limit, your high samples will run through the system as normal, and then the software will automatically run a cleaning cycle so that there's no chance of mercury carryover from your high samples to your low samples. So this just allows the system to be a little more flexible and continue on with the automation. So this can be all defined up front before you begin your analysis. So when you load all your samples, you hit the start button and the system will automatically do this for you. So there's very little hands-on that you have to do during the analysis. As another part of this automated feature, reducing any memory effect or carryover, we keep the spectrophotometer cell preheated. So this keeps any mercury species volatilized. So the problem in some of the conventional techniques where mercury can tend to stick around inside the system, this is designed to keep the mercury from sticking inside the detector cell, so that further reduces and nearly eliminates any memory effect you have from higher concentration samples to lower concentration samples by keeping the mercury completely flushed through the system. Another key feature that the software regulates is the temperature of the decomposition furnace. So there's a thermocouple built into the decomposition furnace that monitors the temperature at 20 times a second. So what this allows you to do is run your fatty or oily tissue samples right alongside your dry soils or plant matter, and the decomposition chamber is heated based on the feedback from this temperature monitoring. So when you in the case of an oily or fatty sample where you may get some burning of the sample inside there, the thermocouple will detect any heat coming from the decomposition itself and slowly regulate that out. So you can predefine this feature prior to your analysis, but generally for the most part you'll only need one or two methods, where you have one method for all of your samples, and then if you are running some very high oil or fat samples, you would have another one that allows you to extend out your heating cycle, and this can all be done on the front end through the software, and this PID controller makes sure that the heating cycle sticks to this user-defined heating profile. One of the other key things to consider is that no matter what system you purchase on the front end, you don't have to know exactly what samples you'll need for the lifetime you own the unit. Any of the configurations can be upgraded or converted over to one another. So if you're at first only running enough samples to justify the dual cell, you have no plans in the foreseeable future that you'll be running anything lower than what you need, but you do have that option to, say, 2 years from now, upgrade to the tri-cell if a project comes up where you need to analyze wastewaters at a very, very low mercury concentration, so you don't need to know exactly what you'll be running at the time you select the unit. So this allows you to be very flexible in what you can consider, because you can convert between the different configurations that we offer. So I spoke before about a carrier gas being used to carry the mercury through the catalyst and into the spectrophotometer cell. So there's two options that we offer for this. One would be pure oxygen, in which case you would use an oxygen tank to use connected to the machine to carry your mercury through the system. We also have an air compressor that can be configured with the system. So this would result in a never-ending supply of carrier gas, because it's going to be compressed air from the environment. But what this also means is that you can now truly bring the system anywhere and not have to worry about having a supply of tank oxygen on site with you. If you have the compressor with you, you'll have a never-ending supply of carrier gas. So it really aids in the mobility of this system and not being confined to a laboratory space. We also offer a gas analysis configuration for the system, and how it generally works is you would flow your gaseous sample through a mass flow controller, which will measure the volume of gas. The gas is passed through a sorbent trap, which collects all of your mercury from your gaseous sample. Then these sorbent traps are loaded onto the sampler tray and are analyzed just like any other of your samples. So once the mercury is collected onto the sorbent traps, it's run through the decomposition furnace. All of the mercury is volatilized and analyzed by the system as normal. So this is not a very common type of application, but we do have more than a few users regularly analyzing gas, so it does have that functionality as well. So at this time, I'm going to turn the slides over to Maggie Castellini, and she's going to tell us a little bit about her lab, what types of samples she works with, and what type of lab environment she has. So Maggie, why don't you tell us a little bit about some of the research that you do and some of the people that you work with in your lab.
We look at contaminants with respect to wildlife health, but also because the wildlife species and fish that we are using are used as subsistence foods and in major commercial fisheries, so they do impact human health as well. Because we're taking this ecosystem approach and we're interested in the flow of mercury through the ecosystem, we tend to study a very wide range of species and also a wide range of tissues, because we're also interested in toxicodistribution within an organism. And so I've listed off here just some examples of the different kinds of animals we've studied in the lab and the different tissues we've measured mercury in, so that you can see what a wide range of tissues you can actually approach with the DMA-80 quite successfully. So for instance, for fish, we've studied a huge range of species, from salmon to halibut. Elasmobranchs, we've studied sharks and rays, which can have quite high mercury concentrations. Those animals are from Mexico. Some marine invertebrates, and in those animals, we tend to focus on tissues like muscle, liver, kidney, and sometimes fish roe. We also study a lot of different types of marine mammals, mercury and marine mammals, and that's because marine mammals are really, really excellent sentinels of marine systems. They tend to be high trophic level feeders, although they're not all, and so they integrate a lot of different nutritional signals, and so they're very, very useful for us when we're considering the whole idea of One Health. In marine mammals, we typically look at hair samples. It's a really good monitoring tool. Also sometimes epidermis, if we're studying whales or dolphins. We use blood samples and blood-soaked filter paper samples. We have a really big project looking at using blood-soaked filter papers to monitor contaminants and disease in wildlife, and so we do a lot of the analysis on the DMA-80 of those samples. In marine mammals, we also look at plasma because we're interested in compartmentalization within the blood. We look at muscle, liver, kidney, and heart. So we're kind of all across the board. The species that we've looked at include a wide range of foetid seals. So in Alaska, that's a lot of the ice seals like bearded seals and ring seals. We've done studies on harbor seals in California and even some Antarctic seals as well. We also study sea lions, including stellar sea lions that are part of that species is endangered in part of their range, fur seals, dolphins, whales, and walrus. We've also done studies on some other mammals, and in those cases we've mostly used hair or blood-soaked filter paper. And some of those species have included caribou, fox, and polar bear. In addition to all that, we've also studied mercury in human hair, and in that case we've actually been able to do segmental analysis of human hair because the DMA-80 allows us to use relatively small sample sizes. So we've been able to look at mercury concentrations along the length of the hair and assess temporal exposure. So you can see that we're dealing with a really wide range of matrices, and the DMA-80 has really allowed us to do that. So looking at our lab structure, we have two main PIs in the lab. Dr. Todd OHara, he is a veterinary toxicologist and pharmacologist, and Lori Rhea, who is a nutritional physiologist, focusing particularly on marine mammals. Then there's me, a research professional. I conduct independent research projects and do analyses related to my own independent projects. In addition to that, though, I also train students to use the DMA-80 and other laboratory equipment for independent research in our lab, and I help students formulate research projects. I also maintain all the laboratory equipment, including 3 DMA-80s. So we have 2 dual-cell DMA-80s and 1 of the tri-cells that Jason was describing. And because the reason we have that is we have enough throughput that we pretty much reserve the tri-cell for samples that are going to be low concentration and try to use samples where we expect concentrations to be higher on the other 2 machines, and that seems to work really pretty well for us. So let me see. I think I've skipped ahead here. I think the slide skipped ahead. In the lab we have, yeah, there we go, thank you. In the lab we currently have 5 active graduate students who are doing research on mercury. They include 2 master's students and 3 PhDs. And then we also have a number of undergraduate students. The undergraduate students are typically mentored by the graduate students and also by myself. And they're hired as student technicians as well. And so they often conduct their own focused research projects. We typically have about 2 to 4 undergraduates per semester in the lab and also during the summer. So we consider ourselves to be a teaching research lab. It's an important part of what we do. We put a lot of emphasis on training and research opportunities for undergraduates. And so that means we've got students teaching students and me teaching the more advanced students. So having an instrument like the DMA-80 that is amenable to that is very useful in our lab. Before we had DMA-80, when we were wanting to look at mercury concentrations, we did use the cold vapor technique that Jason was describing earlier. And as he said, that technique it's a very good technique, but it's quite time and cost intensive. It requires sample digestion, which includes using hazardous materials and having students have to use hazardous materials, and also the waste disposal that's associated with that. It has a relatively low throughput compared to the DMA-80, especially if you don't have an automated system. We had a non-automated system that really required constant attendance, and it is quite complex instrumentation relative to the DMA-80. So it's not really very amenable to being used in undergraduate projects. It's not really, a student's not really able to come in with a 3 or 4 hour block of time and get something meaningful done, whereas the DMA-80 will allow that kind of thing. So switching over to the DMA-80, the first thing that we noticed right away is of course the minimal sample processing. That's really, really convenient and speeds us up a lot. Jason described that. We actually do a little bit of processing. We typically freeze dry most of our tissues except for blood. For two reasons. One of the reasons we do it is because since we're doing stable isotope analysis on these tissues as well, we need to freeze dry them anyway. So it's not an extra step for us, it's just a step we're doing for another process. But in addition to that, it allows us to take a larger piece of tissue, freeze dry it, homogenize it, and that gives us a more representative sample of the tissue as a whole, particularly for certain tissues that don't have mercury distributed homogeneously within them. The tissues like liver, kidney, and hair can be quite variable, and so it's nice to be able to get a homogenized sample of a larger tissue. Having said that, though, the ability to use small sample sizes in the DMA-80 is really great. And if you have limited sample size, it's really good. If you have samples with a high concentration, you can decrease the actual sample that you run and keep it within the calibration range of the machine as well. So, for instance, with hair, we have sometimes run samples as low as 6 milligrams of hair at a time, and it really kind of depends on how good your scale is. And then most of our other tissues, we typically run at about 30 milligrams of dry tissue, since we're mostly doing dry samples. We get very high throughput with the DMA-80. Conservatively, I'd say it's very feasible to get over 20 samples in triplicate run per day per instrument, and then including on top of that a number of QA/QC samples. Our lab is quite rigorous about QA/QC, so with every run we have at least one liquid standard, one certified reference material. Very often we have more than that. If we're looking at a wide range of concentrations, we might use more reference materials that span a wider range. And then about every 20 measurements, we will also rerun liquid standard. So that's a lot of extra samples that we run for QA/QC, but because the throughput of the machine is so high, it really isn't an issue. We can still put through a lot of samples. The other thing that we really like about the DMA-80 is it really does have exceptional recovery and reproducibility. So when we're running certified standards, we're typically looking for getting between a 90 and 110% recovery. That tells us that we're getting, good, reliable results. But on the DMA-80, we usually do actually quite a bit better than that. We usually get between 95 and 105% recovery. And that's consistent over thousands of measurements. In addition to that, we usually have a relatively low relative standard deviation between triplicates, usually, well, definitely below 10%, often below 5%. So we really have a lot of confidence in the data we're getting from it. In addition, as Jason has said, this instrument really does accommodate a wide range of concentrations and tissue types. We've put everything we can think of through it, and we continue to get really good, high-quality results. And we get such differences in our tissue types. We have some tissues that have really high fat content, even if they're, well, even when they're dry, the fat is still there. So, for instance, shark liver is really fatty. And we have other tissues that have very high protein content, like hair. Our concentration ranges of what we're looking at across all the different types of tissues. In something like mammalian plasma, some invertebrates, some low concentration fish mussels, we might have concentrations as low as 5 to 6 parts per billion, but at the same time we might have other samples like marine mammal hair where the samples can actually be 100 parts per million. And even a little higher than that. So, and we're able to accommodate that with a tri-cell and with a dual cell without any problem. So these advantages have really revolutionized mercury analysis in our lab and have broadened the questions that we've been able to ask. Previously, our work tended to center on a question within a single species, single individual species, but now our questions tend to be much more ecosystem-based because we can put through so many more samples, it's inexpensive to run, it's easy to train students on. So that gets us to the next thing about being a teaching research lab. This is, as I said before, we do a lot of teaching in our lab, and we have undergraduates who are mentored by graduate students and me. Those students do a number of different things in the lab. They may be doing an independent study class. They may be doing an honors capstone project. They're sometimes doing internships. The DMA-80 is perfect for this kind of thing because of the ease of use of it and the ease of training. It's really pretty easy to understand how it operates. Jason just went through it with you, and we do the same thing with our students, and it's really pretty straightforward. It's easy for them to grasp exactly what's going on within the machine, and I think they really appreciate that. Some equipment is very complex, and they might get a general overview of how it works, but they might not really understand all the nuts and bolts. But the DMA-80 is really pretty straightforward and pretty easy to understand, and also pretty easy for someone with basic lab skills to be able to operate. So it's usually the first instrument in the lab on which we train students. It also allows students to work in relatively short blocks of time because of the automation, and this accommodates their class schedules. So that gives us a lot more flexibility about what types of students we can bring into our lab. They don't have to have a full day available to come and work in the lab. If they have a 3 or 4 hour blocks of time a couple of times a week, they can get a significant amount of work done. They can either set up samples and then go to a class and come back. There are enough of us in the lab to monitor them, or they can also just set up so that they're only running, 3 or 4 hours worth of samples at a time, and either approach works fine. In addition to that, they get instantaneous results. So they can monitor their QA/QC right from the very beginning and make sure everything's actually working on at any given day. And so that's really nice for them too, because they don't waste time, running a whole set of analysis and then find out later that it didn't work. If there's a problem, they can recognize it right away, come and find me or a graduate student, and we can sort them out. So that's really good. And the really good reproducibility of the data is also great for them. There's much less frustration for the student actually doing the studies, but for us, we can have much more confidence in the data that's being produced by them than we might have if they were using a more complex piece of equipment that might require more tweaking to get good data. So altogether, this makes it really applicable to a wide range of student projects, and we've even done a few high school science fair projects. We've helped students with those as well. So when it comes to the challenges of working in Alaska, the most obvious one is, in fact, the biggest one, and that is that we're pretty remote, and there are relatively few labs working with measuring mercury in Alaska, relatively few labs that have DMA-80. There are some, for sure, but they're not all over the place, and we're far apart from each other. So that means that the cost of service for all of our equipment, not just the DMA-80, for service agreements, for service visits, is extremely high because of the cost of time, travel, and shipping. And so when you've got a lab where you've got multiple high-tech pieces of equipment, that becomes a real financial burden for the lab. And that's something we struggle with all the time, is being able to have enough money to keep reasonable service contracts or service visits for all the equipment we have, not just the DMA-80. And so I think that's something that everybody in Alaska has to deal with. And one of the really nice things about working with Milestone is that they really are, they're really flexible and they're really available. And it's, anytime I've had a question or some issue that I didn't understand what was going on, I could pick up the phone or send an email, and I always get a very prompt response. And then a lot of help with troubleshooting over the phone. And another aspect of flexible service is, for instance, there are other machines in Alaska. So if a service person is coming to Alaska to work on another machine, sometimes we'll coordinate with that and have them come to our lab and do some preventive maintenance, or maybe fix a problem that's just been ongoing with a piece of equipment, something like that. And that can help us defray some of the high cost of travel. The Milestone, though, has great ability to do remote consultation with expert service representatives like Jason. I've actually been on the phone with Jason fixing things, and he's been a huge help. And they also have online support. So that's allowed me to be able to perform maintenance and to troubleshoot and fix a lot of problems without necessarily having to have a service visit. when you have equipment, 3 pieces of equipment for 8 years, inevitably parts are going to fail, things are going to not work. But I've been able to do a lot of replacing basic things just with phone support, and that's been really great for us and for the lab, and it's one of the reasons that we stick with Milestone, because it makes it affordable for us to keep the equipment operating at its maximum potential. And then the other thing I'll just finish up with, I really like the system monitoring software that's on board with the DMA-80 that shows you all the different temperatures that are going on in each compartment and voltages and stuff like that. It's very useful once you're familiar with the equipment to be able to troubleshoot any little things that might start to happen, like if we're getting low recovery or things like that. I can get a much better sense of what's going on with the equipment, know if I need to change consumables, know if there's something else going on with the machine. So it's super helpful for me. And then in addition to that, the software saves all that running information as you run samples, and so that can also be sent to Milestone, and they can take a look at, what your running conditions have been and consult back and forth about, how you might be able to tweak the system to make it work better. So all in all, it's been a really great thing. I mean, we have 3 of them for a reason. We like them, and they're the real workhorses of our lab. We get a huge amount done with them. So that's it for me.
All right, thank you, Maggie, very much. It was very informative and very good information. So I thank you again for helping us out, put this webinar together. I'm just gonna go through a brief summary of what we talked about today, and then we'll open it up to questions from the audience. So Maggie, if you don't mind, if some of the questions come up that I think maybe you might want to answer, if you'll stay on the line here with us, that would be great as well.
Sure, happy to.
So before we get into the questions, just a quick summary of kind of what we talked about today. So again, we're on our 6th generation of DMA-80. So we've developed a very stable and sensitive system centered around our catalyst and amalgamation, the heart of the unit itself. So we've also added some things around this, the software, the temperature monitoring for control over different types of samples, over a huge range of what you might be running, and things like the preheated cuvette, the auto blank, being able to run different types of applications in the same analysis cycle. We've added around, our catalyst and design to enhance its functionality and make it a very useful tool for your lab. So, of course, if anyone would like some extra information, we have a lot available. So we have a lot on our website, if you see the note below, and we'd be happy to provide you with any other information that you can think of, and certainly we're always available for questions here in our office. So now, if anyone has any questions, I will go through. There was a couple that came through towards the end of of Maggie's talk. So I'll start from the first one. Someone had asked if how the reliability of the instrument and how long it would take for a service technician in Suriname. So the unit itself is very reliable. We generally recommend preventative maintenance visits as part of the normal maintenance of the machine, and this just is one of the things that helps keep the unit operational. So out of all, we have over 400 units installed here in the North America, and at any given point in time, 99.9% of them are operational. But again, as Maggie kind of indicated in her talk, a lot of times we can discuss issues remotely without having a service visit scheduled. But again, the machine is very reliable due to it's kind of a simplistic design, so there's not a whole lot to it. So it does result in a very reliable instrument. Someone asked also service in Suriname. So in South America, we actually work with a distributor so we can put you in contact with the information there, but we do work with a third party for South America specifically. And the next question was, how do you conduct the performance qualification on the DMA-80? Maggie, I think this might be a good question for you to jump in on. I'm sure you do a lot of performance qualifications on the system itself, so if you could give us some insight into what you guys do.
Yeah, so I assume that you mean by that whether or not we're getting good data back from the machine. That's what I would call quality assurance, quality control.
That's yeah, that's what I would the question leads me to believe. They didn't delve further into that specifically, but
yeah. Okay, so some of the things we do is we're very careful about how we, well, first of all, we make sure that we run some type of quality control measurements with every single run so that we can really assure from run to run that we're getting good results and consistent results. And so, but the other thing we do with that is when we're starting with new tissue types, we are always trying to find certified reference materials that match fairly closely the type of matrix and also the concentration range that we might expect from our tissue, so that we're trying to run a sample that's similar in makeup, because it's definitely true that samples will burn differently if they're high in oil or if they're dry versus wet. And so we try to make sure that our reference materials match as closely as possible to the tissues that we're running. And then, you know, when we start with a new tissue, we will run extra replicates, making sure that we're getting repeatable results. We have participated in interlab comparisons as well. We were involved in like a 3-year interlab comparison of hair analysis, and those analyses were being done using a variety of methods. And our results with the DMA-80 always came right within, you know, with the means, and there were never any outliers at all. We were always sort of bang on the concentrations that other labs were getting. So yeah, so but with method development, we try to yeah, compare, and we have actually another project where we're comparing our mercury concentrations that we are getting from our DMA-80 with another one in Alaska, and also using the cold vapor technique, and we're actually in the middle of that project as well. So we take a number of different approaches, but we do take it seriously. We don't just put a piece of tissue on and assume that it's going to work. However, it usually does, but we want to be able to actually, you know, be able to publish that we know that the results we're getting are comparable to other labs.
All right, thanks, Maggie. So one question that was from the chat room was how to evaluate the lifetime of the catalyst. Is there a certain test that we recommend to evaluate the lifetime of the catalyst? So it's actually a pretty simple test that we recommend running. So the average lifetime of the catalyst is approximately 6 months, so they're replaced biannually. So what we normally see when we get towards the end of the catalyst is the calibration will sort of no longer be valid. So there's a quick test that we use to evaluate it, and what we do is run 5 replicates of a mercury stock solution of about 10 nanograms through the system, and we evaluate the RSD of those 5 samples. So if those meet our criteria, then it is likely that the catalyst is still within its valid lifetime. But it's kind of one of those things where if you see that it is not kind of performing the same way, running that test will certainly let you know. But on average, we see them about on average about 6 months worth of lifetime. Again, that is very dependent on what sort of samples you're running, how, and what concentrations of those you're running as well.
Can I just throw something in there too, Jason?
Absolutely.
Yeah, one thing that we do in our lab, because as I said, we are running these standards every time we do a run, is we keep a log right by the machine too, and we actually write down the recoveries we get on our standards and the variability on them, and so you can start to see if those recoveries start to fall off.
Thank you. Next question was, what sort of blanks are used in the analysis? Understanding that there's no preparation of the tissue samples, what constitutes a method blank? So when we run a blank in the system, we're actually running the boat itself without sample, and we are running that. So we're looking at the height evaluated through the system. So when you're running the system, the whole system sort of fires up and everything gets hot. So it's going to burn off and volatilize any of your mercury that's through the system. So we have a set instrument specification for what's considered a method blank for the instrument. So before we begin our analysis, we'll run the system to make sure that our blanks are low, and that's by evaluating the peaks through the spectrophotometer. And once we hit this criteria, then we have our blank, our nil value, so we're ready to conduct our analysis afterwards. The next question was, besides a mercury trap, what are the other major maintenance items? And really the only things that need to be changed out on a regular basis on the machine are the catalyst and amalgamator. Again, they're about an average lifetime of 6 months on the catalyst. And then once per year, we recommend changing out the mercury trap, but there's no other parts or consumables that have to be changed on a regular interval. When we do our preventative maintenance visit, the engineer will monitor, we'll check out all the components to make sure that all the parts are functioning normally. But as far as scheduled replaceable items, the only ones that you would be replacing are the trap itself and then the catalyst and amalgamator. So there's a question if the DMA requires a special clean room or can it be placed in a regular room where other trace metals are analyzed. So you do not need to place the DMA in a clean room, as this kind of goes back to the machine blanking. So when you're running your machine blanks, your carrier gas will have any background levels of mercury analyzed as part of the blank. So you don't need to run the system in a clean room, and you're only analyzing for mercury itself. So any other contaminants would be trapped inside the catalyst. The cuvette itself is in a closed system where the carrier gas must pass through the catalyst and amalgamator before the system will analyze. So for that reason, you could just have it in your regular lab space, and you don't need a dedicated space next to a hood because of the trap. So it can just be in the regular lab space, or even on a remote field site if there's a remote lab location that you're using for analysis, it can be placed there as well. Next question was, are there any plans to have a method developed and listed as an ATP for a Clean Water Act using the DMA-80? So at this time, we're always looking to expand into accepted methods for the DMA-80. So I don't specifically know if we are currently applying to have the DMA-80 approved for that specific method. I can get further information for you, and if that's something that you're concerned about, we could certainly work with you to get that information specifically for that analysis itself. Next question was again about the clean room for analyzing mercury. So I'll just reiterate that you do not need to run the DMA-80 in a clean room, and you are still able to get extremely good data. Again, because of this, you're running your blanks and you're analyzing your background before you start your analysis, so you do not need to have a clean room to contain the DMA. And the last question that I see is, how often should a calibration be done when the catalyst is still good? And on average, the machine will need to be calibrated every 3 to 4 weeks or so. However, there is some variability in that average depending on how much you use the system and what types of samples that you're running through. But on average, it's about 3 weeks. So normally how I recommend calibrating the system is with a few serial dilutions of a Mercury liquid standard. So what I use in my lab is a 1,000 ppm stock solution, and then I dilute to 1 ppm and 0.1 ppm, and I calibrate the unit from 0.5 nanograms all the way up to, or 0.5 if you're working on a tri-cell, 1 nanogram if you're working on a dual cell, all the way up to 500 nanograms, depending on how large the sample or how large the concentrations are that you're working with. But I use a liquid standard for that. The good thing about that is they produce a very accurate calibration curve, and they're much cheaper than using your solid SRMs, so which can cost you several $100s per sample vial. And the liquid stock solutions are a fraction of that cost. So Maggie, I have another question from the chat for you, and their question is, would analysis of blood-soaked filter paper be fit for use for human blood samples, and how do you homogenize the paper sample?
Yeah, I think it would be fine for human blood samples, as long as the blood is dry by the time it's on the filter paper, but you would probably still have to have, after analysis cleaning protocols because you're handling human blood. But we don't actually homogenize the filter papers. The filter papers are designed, this is just good luck on our part. They're Nobuto filter papers, and the part of the blood, the paper that gets soaked is exactly the size of one of those little nickel boats. And so we cut the filter paper off of its little holding thing, and we've confirmed that they're also made in a way that they have a totally consistent mass. I mean, we've measured 50 of these in a row, and we periodically will pull a new lot out and measure them, so that this cut-off piece of filter paper with nothing on it always weighs exactly the same amount. So we know how much of the weight that the DMA-80 has on it is filter paper, and how much of the weight is blood. So you can just take that nice dried strip, place it right in a nickel boat, and just combust the whole thing.
Thanks again, Maggie. It looks like that is the last question, excuse me, from the group chat room. So I do have one that was a good question here that was sent to me directly, but what's a good way to tell when to recalibrate the cells, and what problems could occur when using only quartz boats for samples? So the first part of that question is by running a quality control, either SRM or LCS sample to validate your calibration. So when I start my analysis on any given day, I will run several SRMs, matrix match SRMs to validate my calibration to make sure I'm still within range on that. And when your SRMs start to come out of range on your calibration, that's how you would identify when you need to recalibrate the cell. And the second part of that was what problems could occur when using only quartz boats for samples. And there's no difference between running only quartz versus quartz and nickel or just nickel. Again, we recommend the quartz specifically when you're dealing with any sort of acidic type sample, and the reason that is for the corrosion of the nickel boats themselves, but you would get the very reproducible data between the quartz and nickel boats. So we will have the slides available after this presentation. So those will be available. We have everyone's contact information, so we can make this available to you. Maggie, someone was interested if you'd be willing to get in contact with them about some information on the filter papers and the method that you use for, I'm assuming this is back to the blood sample analysis on filter paper.
Yes, absolutely. Do they want to give me an email address or something, or how do you want to do that?
Yeah, I could take that. I think we have a log of everyone's email, so I'll jot a quick note down to myself so I could forward that over to you, Maggie, and get you in contact.
Okay, that would be perfect. Yeah, I'd be happy to do that.
All right, that's all the questions that I have at the moment. If anyone, of course, has any questions after we conclude here today, certainly feel free to contact us. If you just go to our website, milestonesci.com, you have access to some of our emails and also some application notes and stuff like that on the DMA for additional information. Or you can email us with specific questions at applications@milestonesci.com. Okay, so if there's no other questions, I'd just like to, again, thank everyone for joining us here today. Maggie, thank you for your participation. I'm sure everyone found your information very useful and very enlightening. And again, I would like to personally thank you for helping us out here today. So thank you, everyone. Enjoy the rest of your day, and I hope to talk to each and every one of you at some point in the future.
Thank you very much, Jason.
Thanks again, Maggie.